This chapter, we will end with a test, and you will have a quiz. You will have an open note quiz over the material that we're doing. So some people do a good job of taking notes, but over the year I've seen where they take notes. They take notes in several different places. Make sure that you know where they are. Those of you tuning in at home, welcome. We miss you again, Emily.
Yes, Emily.
Wow. Oh, okay, Maddie wasn't so sure, but now we've convinced her that she misses you too. But so I would write down, take some notes. Again, it's just the stuff in red. The rest of the stuff is good. It's important. It's not just advertising, but the stuff that we're going to count as critical will be in red, and this will be on the cell.
She waved it away.
All right, we'll try that. And Erin, can you open at least one of those? Let's see.
None of this.
And
Because you were gone every day, we did those notes. Or you slept.
Adrian, can you turn off the lights and see? Because for videoing,
I'll give it to
it
you.
works better if there's not quite as much. And let's see. Maybe Erin, try the next one too. Open up one more. No, I don't know if that did any difference, but let's see. All right.
You probably have to skip like 2 weeks.
We've been together.
And ladies, you'll be together again all day. So I know. So let's focus on biology now. Mary, your table's not quite straight.
I know, it's irritating.
Yeah, I get it fixed out. All right. So we are gonna. We're going to go into the cell. Hopefully this one's a little bit different than whenever you have studied the cell before. We're going to see a lot of the same organelles inside. We'll talk about the nucleus and the ribosomes. We'll talk about the mitochondria and all those. But again, I will have not done my job that I've set out to if you're not at the end amazed at what's going on inside. And it's easy to think that, oh, this is what's going on inside, a little bit like if you watch that movie that everybody thought was Pixar's pinnacle moment when they did Inside Out and they had all those little people, emotions running around in there. I guess I didn't see it as quite as enlightening. Oh, now I understand my daughters and my wife. No, I still didn't really understand them all that much. But you get the idea of that's what's inside and working, and people get the idea as we go through this that all the stuff that I show you that's actual videos from inside, that's what's happening inside of you. You as in collective big you. But it's not. It's even more amazing, I think, that it's what's happening inside each of the 100 million little component parts of you with inside. So, and usually we start with, at the beginning, if, and I show you, let's say that we're actually going to get back to regular life, pandemic-free, and Emily would be with us on a full-time basis. And we were going to start, we started the OCA kind of to pay for it, a shoe factory. And we've got land somewhere, so we have acres and acres of land, but it's up to this class, these 14 students that are in here, to come up with what we need to start this shoe factory. Let's make it an Adidas factory, since Nebraska is an Adidas school. We'll be close by. We have a big customer. So just raise your hand. What are some of the things? Yep, right there. That's fine. Thanks. What are some of the things we would need in a factory? Okay, so we're gonna have to have something that actually builds it, the production thing, the machines. We're gonna need employees, which some of you might thinking, hey, great, I can get a job here, I set the place up, I've gotta have a place in. We need employees.
Probably need product.
We need products, we're gonna have to have something, and the products, you can't just send out, like just kicking out shoes and throw them into the back of a truck. The shoes all have to be in a box, some kind of packaging that's in there, and it's gotta be labeled to say what's inside, and then all of those will go inside probably another box and say where they're going to, so that they know, okay, this is what Shields ordered, this is whatever the Foot Locker ordered, whatever, they're gonna have to have that. What else are we gonna have to have? Okay, you're gonna have some kind of currency, you're gonna have that, you're gonna have management,
A bill.
you have to have a building.
Good infrastructure.
Okay, that's true. What would that entail?
A system.
That sounds like somebody who's ready to be upper management right away. What are we gonna need in the infrastructure?
You need a solid foundation.
Oh my word.
All right, now I'm thinking of a hymn, and I'm ready to sing "How Firm a Foundation," but keep thinking about that. What are we
Like
gonna?
a system of like how everything's gonna be made.
Okay, so you have to have like an assembly line or something, and even that, just a way for our Simon's workers to get around. The last part of college, I worked at UPS, and they used to give tours. You know, post 9/11, not as many places give tours anymore. But when you go in there, it's an amazing place to just walk in, and you see catwalks everywhere where people can get from one side to the other without going down, and there's forklifts and stuff moving all over the place. You just walk up over the top and you can go straight. You almost need a map to get there, but then there's also belts everywhere because the boxes go up and over. When you walk in, it's a little bit like Monsters Inc., and you see all those doors going everywhere. There's boxes that are going just like that, and they get to a certain spot, and then they get divided depending on what a sorter pushes, what button it pushes, and this one's going to this gate over here because it's all the ones that are going to Denver go to there. We're going to have a full trailer load of stuff going to Denver, so we'll send a pure Denver one. And you just watch the boxes. Now, I worked on a slide for East Omaha, and you looked up and you wanted to see just kind of a line of boxes coming, keep you busy, whatever. What you didn't want to see was just a crush of boxes coming when they opened a pure Omaha trailer and they came down. But all of that stuff, we would need something like that, which takes a lot of work, where everything can be sorted, it can go, and it doesn't have to be down on the ground where everything is all congested. And you can't see past anything because there's all kinds of stuff, and then you just hear the beep beep of all these vehicles going around. You wouldn't get anything anywhere, so they just lift everything up, and all the boxes, all the people are up there. It's a great place to work. Part-time, I had full medical, dental, vision, which somebody who's in college usually doesn't get sick. They don't use any of it, but it's nice to have. They paid vacation, everything. It was a great place to work. The hard part was they expected an awful lot of you. So those catwalks were kind of cool. We couldn't go up there. You had to be a management to go up there. And a lot of times, all of a sudden, you'd see your manager up there with a stopwatch and a clicker. And you have no idea who they're looking at, but they're timing somebody. And you had to do something like 650 packages an hour, sort them with like 98% accuracy or something, and you had no idea, but now all of a sudden it got a little warmer in the building and stuff. So the catwalks weren't that, but that's another thing that we would need would be supervisors. So we've got Simon's employees, but then we'll also need, over the employees, we're going to need some supervisors, the management part that's actually making sure that they're doing things right. And just like everywhere else, we would need security. These days, everything needs security. You're going to have ways of getting raw materials in what you're going to make the shoes out of. You're going to have waste that goes out. You're going to have to get rid of the garbage. All of those things, a lot more. All of those things need for the factory to run. And they're not going to happen by accident. You guys are going to be very busy coming up with how are we going to do this. Planning a factory takes a long time. It's not something you sit down in a weekend and say, hey, let's build a factory. I mean, it's months and months and months of planning and drawing and stuff before it gets built. And all of that stuff that you said and that I added that we needed is all in every cell. And yet, it happened by accident. It's just nice that we could add one thing at a time and somehow the cell just got better and better, and now it works like this, like a factory. So as we go through, you're going to see, hopefully, you're going to see that each cell is like a factory. And we're going to talk about, it'll get better here in just a minute. What we're going to produce is proteins. I mean, that's the building block of everything that we do is protein. So we're going to go through and we're going to say that we're building protein, so that's going to be, each cell is going to be a protein factory. Now they do other things as well, but we're going to talk about that that's what it's going to do. And hopefully, if I do my job correctly when you're done, you will be, wow, that is just like a factory, and you would be hard-pressed to think that this could happen just by accident. If you can come out of this and think, well, no, you know, nature could just do this, then I didn't do my job very well. All right, we're going to start out with some basics. And again, it's not red, so go ahead, Maddie, I'll talk slow so you can get it all down. But you don't have to. All of this will
Wait, so
will come through.
Will not be on the test.
Right. That will not. Red stuff is what you need, but that doesn't mean snooze time. But cells are extremely diverse, and that again, they'll come up several times. That when we talk about stem cells, you hear about stem cells. Stem cells are cells that haven't diversified yet. They can be anything. They will be anything. You started out, all of you started out as one cell. And that cell became everything in you. Now eventually, they specialized. And you've got muscle cell and bone cell and organ cells and all kinds of different cells that specialize, but they all started from one cell and then from just this few cells that weren't specialized at all, and they turned into everything. That's the power that they think of stem cells. That if we use these cells, they can be anything. If we put them in somewhere where those cells aren't reproducing, like nervous tissue, somebody gets a spinal cord injury or a brain injury or a stroke or something, brain cells, spinal cord cells don't reproduce. If we put some stem cells in here, then maybe they'll turn in and they'll make the connections that were broken. That's the hope, that's the thought that they can do. But eventually they do diversify. Most of them require a microscope to see. Human cells don't, not all of them do, but even one that you could see with the naked eye, unless you stain it and make it something else, you wouldn't see it because it's not going to be colored. Anybody know what the only cell that we could see with the human eye is? Some of you probably have enjoyed some other organisms' largest cell as well. It's the egg cell. The egg cell is the largest. Now, the human egg cell, we all started out as, it's about in a textbook, if you look at the end of the sentence and you see the punctuation there, a period, it's about that size. But it won't be dark black on a white background, so you wouldn't really be able to see it. But an egg, any egg, a chicken egg, is a single cell. Largest cell that everything else in the chicken you can't see without a microscope. Largest cell known to man is an ostrich egg. So, but all the rest of them are really small, and they're specialized. Light microscope, we kind of went through kind of who started it. I don't know. Leeuwenhoek gets credit. Some other people do as well, but they're limited by the properties of light. Light has a certain wavelength, and when you get down to something too small, the wavelength is too big. So then we use electron microscope because its wavelength, its frequency is much, much smaller, so we can see more. But we've about exhausted what it can see, and so they're actually looking for something else that will vibrate. Can we actually somehow use a quark that vibrates even faster, higher frequency? Can we look at something using those, something smaller, before we can see that? Did I talk long
Yes.
enough? All right. All right, some examples. Lymphocytes. It helps kill bacteria. Here is an electron microscope. We are not ever going to have a set of 5 or 6 of them sitting up here of electron microscopes. We don't have room. We definitely don't have the funding. We would have to have a whole calendar worth of politicians coming in and speaking to get people to donate enough money that we could get electron microscope. I have never had a chance to look through one. I've seen electron. This is electron micrograph of the lymphocyte. I've never seen one. I've seen pictures, but I've never actually looked through one. I would like to sometime, but I haven't. Light microscopes, and this one is different than ours. It's actually binocular rather than monocular, and everybody says, oh, we should have those. And if you get to college and your college biology might have one because they look so advanced, they're harder to look through because, one, you see twice as many eyelashes. Some of you have a chance to see eyelashes when you're looking through, and it's really hard to focus. Most students end up using these with one eye closed because it's just easier just to look through one than to try and focus through 2 of them at the same time. But otherwise, it's just like ours. It has 4 instead of our 3, and again, we talked about that. The last one is the oil immersion one that almost nobody uses. It's quite a bit stronger, but it's a lot more difficult to use and harder to focus. This is a light microscope. That's what it's talking about. And this is a euglena. We saw some of those, the green, and they're the interesting ones that have the chloroplast, just like plants do. This is a scanning electron microscope, micrograph of a spiderling. That's those things that no matter what you do, there's no way, sorry Olivia, they're on your pillow every night or whatever, and as you breathe in a bunch of them. And
No. You serious?
yeah, but you think that's bad for you. Think about them. That they go to school and they see pictures of us and they say these are the monsters that eat us every night, and there's nothing you can do about it. But they are now. This is tiny. If this is a scanning electron microscope, so this is tiny, tiny, tiny. That's probably magnified like 30,000 times. But look at just the design, how they, I mean, it looks like, yeah, the guy who's in charge, I forget his name in Monsters, Inc., the guy who's,
Waterloo?
yeah, it's water something, yeah. But it was something where it wasn't like Waterman or water, it was Water Noose. Was it
It
Water
was some
Noose?
sort of name that someone made fun of.
I don't know.
At some point.
Yeah. Emily at home, you've got time, look it up. Let somebody in here know. All right, another picture, and again, here is to scale human egg. So this would be the size of textbook. Open it up at the end of you reading the, oh no, I forgot what it was, that the window or whatever, what was the literature thing you guys, the ghost or the shadow or
It was the window.
window.
Cottage in the dark souls.
The black cottage, like
Dark
that.
souls.
Okay, whatever, you're reading your book and you get to the end of a sentence, and the period at the end of that sentence, that's about how big. You can use that as scale. So this is blown up that much. And not everything is to scale. Obviously, a blue whale is much more than I mean, that looks like maybe 4, 5 of this human. Well, blue whales are much, much longer than what that is. Those aren't all drawn exactly to scale, but everything from here, you need a microscope for. The light microscope is in the blue, so we can see this stuff. Now, we aren't going to be able to see that much detail inside. We will see chloroplasts, but we won't be able to see that much detail inside. That's the electron microscope is the kind of pink here that makes this, I don't know, is that mauve? That color, the pink and the blue together. That's a scanning electron micrograph of what a chloroplast is. We can see these, you'll be able to see cells, you aren't going to be able to see everything in there. And viruses, nasty, but they look like space aliens that come down, drill a hole coming down from here, and then they shoot their DNA into the cell that takes over the cell. This is what our factory is going to be making, proteins, and that shape is important. It's not just the order of that. I know it looks kind of like one of those balloons where they twist them into different shapes. But the shape is important. If it's the wrong shape, then it won't function. It's said to be denatured. Amino acids are huge. That's what the proteins are made of, huge molecules. We aren't quite down to atoms, and that's why the most curious of scientists are working on something, what else can we do rather than an electron, and get down so that we can actually see the atom, so that our model of the atom can either be confirmed or it can be brought into focus and changed a little bit. Obviously the stuff on the other side is all made of cells, except the frog egg. Frog egg is still just one cell, and those you can see. If you've been to a pond, you might not know what you're thinking, oh, what's that scum or whatever. Sometimes it's just a whole jelly-like ball of it, and they have to lay a whole bunch of them because most fish and aquatic animals find them somewhat tasty, so they have to lay 1,000 so that whatever 150 will actually hatch, and then out of those something will eat a whole bunch of the tadpoles, so a few frogs will be left. Otherwise it would be overrun. It would be plague-like with frogs if all of the frog eggs hatched. All right, oh, that's what Colton was waiting for. Finally some red. Why are cells so small? In the next one we will make this make sense. I will do a couple of drawings to try and show you what it's like. And they need the surface area large enough for entry and exit of materials. Our factory for making shoes would have roll-up doors where semis could back up to, and we could get stuff in and we could load stuff out. The cell is actually a little bit more efficient. Everything can kind of come and go from the cell almost anywhere, but you have to have enough room to get stuff in and out. The bigger the cell gets, the more door space you need to get materials in. There's no reason to have a huge factory if you only have one little door and you can't get enough stuff in to actually use all the space. You could have just had a tiny little strip mall factory with all the stuff that you could get in. You'd be able to keep up. So it's the surface area to volume ratio, and you can think of it as the door to factory ratio. How much stuff can we get in and out compared to how much space there is to actually make whatever we're making. And small cells have more surface area for exchange. They actually have less surface area, but more, the ratio is greater. And there will be a picture. The adaptations to increase surface area. Your small intestine is a thin tube, and if you kind of want to know, well, how big around is my small intestine, which is a question that most of us are going to wonder at some point. I wonder how big around my small intestine is. And it's easy to say because if you get natural casing wieners, you get a hot dog with that, you cook them up over the grill and they have that satisfying snap when you bite into them because they're natural casing. That natural casing is a nice way of saying we stuff this stuff into, usually it's either pig or sheep intestine, but ours are really close to the same size. Ours are about the same size. So your small intestine is about as big around as what a brat is, a sausage. It's longer, but it would have to be, to get all of the stuff that we need out of it, it would have to be like probably 10, 20 times longer, which if you picture somebody else, don't picture yourself because that would be horrible, but picture me since I'm standing up here with an extra 10 times as much room in my midsection as what I have now. Not as pleasing a sight, and pants wouldn't fit very well, so God moved it all down by making these microvilli, and you'll see a little bit where it's just little finger-like projections that come out into the small intestine, a lot more surface area there. They do say that if you took, this is not a great picture to think, but if you took your small intestine out, it would probably be somewhere from me to Colton would be a distance it would go if you stretched it out. But if you took and you spread all of those microvilli out and just kept spreading it, you could probably carpet your bedroom with just your small intestine. Yeah, not something you'd want to do, but it will stretch out that much surface area.
Is it that thing that loops up like?
Yes, that thing that loops around, and it looks like it's just like somebody just stuffed it in there.
Yeah.
But your loops are exactly the same as her loops, the same as her loops, so they know, and they have names for each one of the loops. So everybody's is the same, which seems like that's more design than just chance. All right, we'll pick it up from here tomorrow.
You should go over the thou shalt not lie.
Yeah. We have.
This isn't a lie.
All right, yesterday we got to this point where we said that smaller cells are more efficient, and you copied this down. So it was red, you got this yesterday, and that is green. So now I'm going to go ahead and we're going to draw a couple of pictures, and I'm going to show you, illustrate how the surface area to volume ratio changes with size. So,
Not working.
no, wait for it. You have no patience.
It's
All
not
right.
a skill of mine.
Oh no, I don't have that shot. This is the other one. So if I do we don't want to stress your math out here, so I'll try and use fairly easy
How
numbers.
did you do that?
We are going to do square cells only because the math is easier. Does anybody know if I tried to draw a sphere? Anybody know what the formula for the volume of a sphere is?
I forgot also.
4/3 pi r cubed.
4/3.
Yeah.
CJ says that all the time.
1.33 pi r cubed if you'd rather that. But that's a tougher one to do. What's the formula for a cube?
Like base. Oh no.
I remember the guy that taught you this last year. He'd be really disappointed if you didn't know the formula for the volume of a cube.
Did you teach us
I did.
Yeah.
Perimeter. Top.
I'm sorry.
Length times width times height.
Oh.
Oh,
yeah.
Pretty simple.
I feel bad for Chloe too. She taught us that too.
All right, well, we all try. And let's make this simple, because I'm not all that encouraged right now. Let's say that each side of this cube is 1 centimeter.
I'm very good at drawing cubes.
So let's say for this one, the volume is going to be, with courage, say it.
I don't want to say with courage.
Wait.
What is,
What?
what is the
What?
volume of this cube?
1
One what?
Centimeter?
centimeter.
That's a distance about this much.
Squared.
That's an area.
Um,
4/3 pi r
Squared.
cubed.
Cubed.
That's it. Volume is always cubed. One
What's the difference between squared and cube?
squared is
Oh, never
what
mind.
we're gonna do next. It's just the surface area of just this. Cubed means you multiply this, so it's what the volume, all of that space inside can be. But that's a good segue into the next one, because the other part that we said in the last slide was it's the surface area to volume, so we need the surface, whoops, let me just actually write the first one out. I could erase it, but it's easier to scribble. And I am not OCD at all, so, whoops, area. What is the surface area of that cube? All of the area on the outside of it.
That's addition, right?
Yeah, well, part of it is addition. Not really, but could be.
4.
6.
Bruh, I don't know.
She's right. 6 what?
Centimeters.
That's about this far.
Squared.
Squared.
Oh,
6
squared.
centimeters squared. Each one of them is 1 centimeter squared, because it's 1 centimeter on a side, so centimeter times centimeter. But there are 6 sides. How do you know there's 6 sides? Well, yeah, I mean, you can count them, but it's also like a dice. If you roll a dice, it has 1 through 6, because there are 6 sides on a cube. So our surface area to volume ratio is 6 to 1. So now, I'm a little worried about this, but we're going to give it a shot.
We've done this. Kind of. Oh, you so good at drawing.
Make it a bigger square.
Cube, yeah, it's going to come off here, and it's not very cube anymore. I worry about this, but we assume that it's still a cube. 2 centimeters on a side, 2 centimeters on a side, still not scary numbers. We can do 2s. What is the volume of this cube?
2 centimeters cubed. But you have to do
Length times
here.
width times height.
Wait.
6.
No, 8. It's
8.
Yeah.
2 times 3 is 6, but 2 times 2 times 2 is 8 centimeters cubed. And what is, so there's a lot more volume. What is the surface area of this?
Oh, 16
12?
centimeters?
12.
No.
36.
Wait, what's 8? 12?
No, I
Guys
said
are
12
scaring
and you
me.
noped it.
9.
On each side,
There's 6 sides.
but on each side, what's the surface area of each side?
2 centimeters.
4 centimeters
Why is
squared,
it
2
4?
times 2, because it's side times side.
Oh my word.
So 4 times 6, flashcards.
24.
You're killing me. 24.
Here, ignore what I said.
All
What
right.
did you say?
Nothing.
She said, please, Mr. Schmidt, can you get me some flashcards for Christmas? And we actually just went through, we were going to take them to the Goodwill, the flashcards we use with our kids. I will pull them out of the Goodwill pile, and Merry Christmas.
My uncle was making fun of me for not knowing.
Okay, and this is, it's getting a little chilly, but I'll let you, a little history for you with my kids. They didn't like the flashcards. My brothers and I did. We didn't really, but my mom said, as soon as she said, I'll keep score, then we were all in, because we were super competitive.
I hate flashcards.
Right, so what I did, my kids didn't either. So what I did with them was, we sat outside, I sat in a lawn chair, they stood in front of me, and I flipped them, and every time they got one wrong, I shot them with a squirt gun.
Ah.
Why can't you
that's
just give
fine.
candy for everyone who got right?
No, it gets
Ooh,
better.
It's summer.
If they went through all of them, got them right, they had a squirt gun and they got to shoot me. It was called Flash and Splash. Introduce Flash and Splash. Oh, don't kid yourself. When you were mom, you were going to remember this, and you will do Flash and Splash. You won't remember my name. You won't remember who I am, but there was some teacher that said that. Kids love it. They will ask to go out and do flashcards.
I think I'll just stick to candy. That's.
You got a whole generation of obese young people that at least know their math facts.
Yeah,
maybe.
yeah,
Okay.
yeah.
Anyway, surface area, flash and splash. Write it down. Write it down. You're gonna want it. Flash and splash. What is the surface area to volume ratio reduced to lowest terms?
8, 20. Wait, oh.
24 to 8, which is
3 to 1.
3 to 1.
3 to 1.
So by getting larger, here we had a 6 to 1 ratio. Now we only have a 3 to 1 ratio. There's room inside here. Let's go back to the factory thing. We're making something. It's going to be proteins for the cell. But whether it's shoes that we were talking about, whatever, you've got lots of machines in here. You've got a lot more machines. We now have 8 times as much space in there to put machines in. But you don't have as much of a ratio. There's not enough room on the outside to get all of the stuff in and the finished shoes out, in this case proteins out, to do that. So it's really inefficient. So it's much more efficient if we divide this. That wasn't so good. Into a bunch of smaller cells, and so cells keep dividing, because when they divide, and we'll go through that in another chapter called mitosis, we get much more efficient little cells once we divide them, because the ratio goes down. So why do cells stay small? Because they're much, much more efficient. All right, enough of math class for today. What hour do you guys have math? 5th. I'll pray for you guys.
Thank you.
Oops. All right, 2 main types of cells, and we're going to go through this, and then we're actually going to start going through the different parts of cells. 2 main types of cells and the cell theory. Now, you saw in the little cartoon about the cell theory, those 2 guys that went through kind of helped each other come up with it. One was looking at plants, the other one was looking at animals. And they came up with the cell theory, although they had a little bit of problem with the second point, and it started another round of violence in the science world. But the cell theory, some people say there's, I like to say there's just 2. It makes it simpler. All organisms, now read that, organism. What is an organism? Organism, what is an organism? It could be a plant, could be an animal,
Anything that
A thing composed of cells.
anything that is living is called an organism.
I said that, then you can word me.
What?
I said that, then you can word me.
What?
Wow!
So on the test, when you're asked cell theory, I don't want to see, and this is a preemptive strike, because most teachers do not want to actually use their red pen or purple pen or pink pen, if they still have one that's working, to mark things wrong. We would love to give you all 100. And if you make a silly mistake here or there, you get a 98, a 96, that's fine. We aren't in this to, I can trick them into doing this wrong. So I don't want to see everything is made of cells. Her water bottle is not made of cells. It's not alive. All living things are. So they're saying organisms, or you could say all living things are made of cells, and cells only come from pre-existing cells. Now, the one scientist that we saw earlier thought that they spontaneously generated. They just kind of popped into existence, but now we have found that no, there's no way to make cells just come from nothing. That's a problem for both worldviews, as far as creation or evolution, because both of them have to violate that second one. Creation gives a mechanism for violating that. God created cells. He created things with cells. He didn't start with just, I'm going to make a cell and see what happens with that, see if it turns into anything else. But it is a problem with, if this is a law, then at one point nature had to violate a natural law, which is more difficult. Something supernatural can violate a natural law. That's what makes it supernatural. It's outside of the realm of just the nature, so it can. But it is. Both of them do violate that. All cells have a plasma membrane. We're going to talk about that. That's the first part of the cell we're going to talk about, so you'll get plenty on the plasma membrane. Thus, we left it in its original color. Two main types of cells. This has become very important. It's one of the things that divides those kingdoms that we went through. And it has to do with the genetic material, which whenever you see genetic material, that means DNA, which stands for, nice, deoxyribonucleic acid. If you were playing with Madison, she would have given you some candy. I would have just given you the squirt gun and you could have shot me. I don't know which is the big, well, no, squirt guns rarely go pow-pow.
But I go pow-pow.
The two types are prokaryotic and eukaryotic. I'm going to step across the hall and be an English teacher for just a moment. Just a moment. I like being on this side of the hallway. The two prefixes, pro and eu. Pro means if something is a prototype. What does that mean? That's before. That's the first. Okay, so this is kind of an evolutionary term. They think that the prokaryotic cells, although they're still having a hard time finding it, but these are the early ones. If you're going to build a new car, if you're going to build a new airplane, they come out with a prototype first, and they make sure that it works. And a lot of times the finished product doesn't really look a lot like the prototype. It's just where they kind of figure out what needs to happen to do that, and then they perfect it, and then they put it into a usable, marketable version of it. So pro means early or first, and eu means true. It's a prefix that means true. So if something is the actual thing, then it is. So this was the early cells, and once natural selection got it figured out, they became the true cells. So prokaryotic and eukaryotic, just it is evolutionary, but we still use it as terms. We're not going to have our own nomenclature, but that's what they mean. And prokaryotic, and they'll show you a picture here in a second, don't have a nucleus. You know that from what you've studied before in the past, cells, inside the nucleus, you've got all the DNA, kind of as a package, and that's the eukaryotic cells have the DNA inside a package. The prokaryotic, the DNA is just kind of spread throughout. A lot more chances of mistakes, bacteria and stuff. That's why bacteria can change so quickly. That antibiotic-resistant bacteria you studied about last year that seem to show evolution, although they don't really work as well except when that drug is there. If you remember back then, it was, you guys were always beating me to wherever the food was dropped in here, Hunger Games style, but I had when the floor got heated, and I had the carbon fiber wheelchair, and you guys were all melting because of the floor, I could still slowly make my way through, and I got the food, and I was a much better when the floor was superheated. But as soon as you cooled the floor down again, took the drug away, then you guys were still better at it than I was, and I would die out. So that's why that changes so quickly, because they don't have the DNA impact in anything, so it's much easier to make mistakes. I think you just get pictures. One, This is like an animal cell. Relative sizes, this is a eukaryotic cell. They come in different sizes, but an average size. Here's a prokaryotic cell, much, much smaller than that. Here's the nucleus, and all of this kind of blue stuff inside here would be the chromatin, the DNA that's in there. We're going to use different terms for the DNA. It can be chromatin, it can be chromosomes, it can be genes, it can be DNA. Those are all different terms for the same. Depends on what form they're in, but they're all the same material. And in here there is no nucleus here at all. It's just DNA is just in there somewhere. So here is just all of the DNA is just around. There's not any particular place it is. It's just kind of all the way through, so that when this divides, we hope that it all gets put into the right place and each side gets what it needs, but it doesn't always happen. But they're bacteria, they don't really complain, they just go ahead and do their things, and most of them are actually beneficial, they help us. There's a few of them that make us sick and do bad things, but most of them we actually need. All right. So the first one, now we're going to start to go through the different, what are called organelles. Organelle, kind of like an organ, but it's hard to have an organ when we're talking about just one cell, because remember the hierarchy was that cells are made out of compounds, but then we have cells, and cells working together make up tissues, tissues working together make up organs. So there's lots and lots of cells and organs. So in each cell, we have these little things that kind of function like organs, so we're going to call them organelles, little organs, organ-like things. The first one's plasma membrane, and most of the time people think of it as that's the part around the cell, and we think of it kind of like a balloon enclosing it, so that like if you poked it, then the whole cell would just explode or would kind of pop and everything would leak out. That's not the way. Plasma membrane is put together much, much better than that. The plasma membrane is the boundary, separates the inside from the outside. Necessary. But it also does an awful lot of stuff while it's doing that. We'll understand this later. It's going to be really important, and not only with the plasma membrane, but with what surrounds a lot of the other things inside. Plasma membrane is a phospholipid bilayer. So that's an important term. It would also be phospholipid would be a fantastic Scrabble word. I'm not sure how you could make it. You only get 7 tiles, and you got to build on to something. You could do lipid. You can't do pholipid and then phospholipid. You'd have to somehow come up with phospho. Well, I guess you could. 7, you could do lipid and then phospholipid. Keep that in mind next time you're playing Scrabble. That's for when you get stuck with all the vowels and you just don't know what to do with them. Look for phospholipid. The polar heads. Now, we haven't talked about this. It's a chemistry thing, and I don't really, at some point I will need that. But a magnet has poles. We call them north and south. They're actually, we have shortened them down. The North Pole is actually the South Pole, because the name for the North Pole, the proper name is actually the north-seeking pole, And which pole would actually seek the north? It would be the south. So the north-seeking pole and south-seeking pole, we just call them north and south because it doesn't really matter. They could have called it east and west. Well, here in our planet it's kind of north and south, but any opposites they could have called them. Polar just means that it has two sides. Water is very polar. It has a positive and negative. Because of the shape, it's in this little bent. It's a very simple molecule, but because of the bent shape, it makes it very, very important. And life isn't possible without it. So when they look for life in outer space, the first thing they look for is water. Just most simple compound, I suppose besides oxygen that we need. That has only 2, this has 3. Very simple thing, but it's very powerful because it has a positive side and a negative side. And if it does, that means that it can attract other things that are around. It can attract them that side. But on the other side, you try and put it pushes it to the side. It repels them, and they don't want to go together. So these are polar. So the heads are polar, and they point inside and outside of the cell. Gluten free menu and my observations. The non-polar tails. Some things, for example, the pen you're using, unless it's a very special pen, is non-magnetic. It doesn't have any poles on it. That's what the tails are, and they face inward. The important thing about polar substances and non-polar substances are like dissolves like. So polar things will dissolve polar things. Most things are polar, so water dissolves an awful lot of things. Grease and oil is non-polar, so you have to have a different substance to actually wash your hands if you have grease on them. Most soaps have a non-polar part to it, just like this. But like butter is non-polar. But you got to have something in there in your soap that is also polar, because otherwise it would be like I got oil on my hands, and so you take a stick of butter and wipe it and you clean your hand with a stick of butter. But now I got butter all off, and you can't wash that under the water. Water's not going to take it off. So soap actually has a polar part and a non-polar part. The non-polar part sticks to the grease and oil, takes it off, and the polar part lets the water wash it away. So you got to have both with there. These have both, and they hook together in a certain way. And sometimes we need, and this is a word that most of us, bad cholesterol. Again, cholesterol can be very good when you go to the doctor. When you get to be a little older, most people your age, they don't really worry about cholesterol that much because it doesn't become a problem until later. There's 2 types. There's good cholesterol, bad cholesterol, and sometimes you get in trouble because you have too much bad cholesterol. You also can get in trouble if you don't have enough good cholesterol. You need to eat more fish. You need to exercise. That helps the good cholesterol have it. So that's why most of you are fairly active. You get to be my age and things slow down because you're busy with other stuff. It's harder to stay active. So the bad cholesterol goes up, good cholesterol goes down. But in this case, it's support. I thought it was going to give you a picture. It won't. We'll actually end with this. I'll let you, and then we'll stop at this part. Fluid mosaic model. This is what's there instead of the balloon idea. There's a balloon stretched outside, and you can pop it. This is much more creative the way God did this. Fluid mosaic model describes the plasma membrane as a phospholipid bilayer in which proteins are embedded, like little rafts that are floating around doing their jobs. In fact, they actually called them lipid rafts that are made in there, and they often have the different functional things in there, the things for communication, for receiving and sending.